Assalamu alaikum, and a very warm welcome to everyone joining us from Pakistan and around the world. My name is Ras, and on the behalf of Zynome Scientific, I am truly excited to have you with us from this 2-day intensive workshop from DNA to gene expression, a practical journey through the core molecular biology. Before we begin, I want you to take a moment and think about this. Every groundbreaking discovery in genetics, every diagnostic test, every therapeutic innovation starts with a single experiment in a molecular biology lab. And today you are taking a step into that journey. What you are about to experience today. This is not just a lecture, this is not just a theory, this is a complete practical roadmap, the exact workflow that real labs follow every single day. By the time you finish today's session, you will have a crystal clear understanding of how DNA is actually extracted from cells and tissue, how the purity and concentration are measured using nanodrops, spectrophotometers, how agarose gel electrophoresis separates DNA fragments, etc. These are the techniques that form the heartbeat of the molecular biology. If you master this, you can work confidently in almost any genetics, biotechnology, microbiology, or research lab in the world. Okay, now I'm going to share you the screen, so we will start. Anyone have a question? Kindly ask. If you have any question, you can ask me in the chat box. Okay, now start. Okay, today's this workshop is on molecular biology techniques from DNA to gene. Okay, so we are in the first day of the workshop. It is a practical journey through the core biology. Okay, now first of all, I'm going to introduce myself. My name is Ras. I am a BS Genetics and Molecular Biology scholar at University of Health Sciences. Brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, molecular biology techniques, genomics, genetics, and evolution, molecular diagnostics. With a keen interest in advancing scientific discovery, Ali is dedicated to inspiring others through engaging learning and fostering a deep understanding of cutting-edge molecular biology techniques. Okay, now we're going to tell you the aim of the workshop, what you will gain. First of all, it's a comprehensive understanding of the molecular biology technique. Okay, from DNA to proteins. Actually, molecular biology is a, is all about the central dogma. Okay, it is all about the central dogma. it, it is actually the thing in which the information is transferred from DNA to RNA and then into proteins. Okay, then proteins. So we all study about DNA, RNA, and proteins. Can we collect the sample and then how we extract the DNA or RNA or the protein, and then how can we interpret the result? It's all about molecular biology. Tell me if you don't see the screen. Okay, then we get the hands-on knowledge. Okay, the hands-on experience. It is all about the handling. Okay, if you have a good handling, you are the expert of molecular biology. If your handling is poor, your result will be just washed off and you get the poor result. Okay, so understand the essential techniques like nucleic acid extraction, quantification, PCR, and their real world applications. We'll talk about it. Okay. And the third one is the skill enhancement. Equip yourself with the knowledge and skills to excel in research and academic pursuit. Unlock the potential of molecular biology to shape your future in science. So now we talk about the workflow of molecular biology techniques. Okay. So this is a 2 day workshop. So we will talk about first of all the sample collection in our first day. Okay. In our first day, and that things are second. So we talk about the sample collection, then we talk about the biggest extraction, in which we talk about the RNA extraction and DNA extraction, okay, and the chemicals used in DNA and RNA extraction, and what is actually the role of that chemical in extraction. And then we talk about the quantification, in which we use electrophoresis gel. And the second option we have is spectrophotometric, that is the nano. So both have advantages and disadvantages. If we go with gel, they give us and tells us what is actually the DNA. Is it DNA present or not? But in nano drop, we get the quantity. But if the DNA is degraded, then so okay, Cyrus, you don't see the screen. I will share you to you again after stop sharing and then again. Tell me if you see the screen. Okay, Cyrus, now tell me, do you see the screen? Okay, now we are going. to the molecular biology. First of all, the equipment we use in molecular biology lab, I am going to share you some famous molecular biology labs equipment. Okay, first of all, the main thing we use is sorry, the pipettes. Okay, the micropipette. We use three types of micropipette. First one is the P10, and then the P100 or P1000 and P1000. And in the bottom we use different type of tips. For P10 mostly we use the white tips. For P100 and P200 we use yellow tips. Above the P20, above the 20 micrometer we use yellow tips, and for P1000 we use blue tips. So we use the bucket swabs, the mouth swab for sample collection. For the sample collection, different types of things which we use the bucket swab, and we use the tubes, the additive tubes, and the and the sorry, and the anti-coagulant tubes. Okay? And also we use the thermocycler. Mm, Thermocycler actually is uh, used to amplify the gene of interest, and we do for PCR. The mostly we use for PCR. We can also use to incubate and for making the cDNA from RNA. For RNA to cDNA, we can incubate the RNA into thermocycle. And we also use the appendrops, the different types of appendrops, the 50 microliter appendrops, the 50 milliliter appendrops, the 1000, and the different types of in animals. Okay, and we also use the different type of chemicals in molecular biology, like this type of things, like phenol, ethanol, propanol, glycine, acetic acid, that type of molecular biology chemicals we use. Okay, and the next one is centrifuge, which we use to homogenize our sample, or to use for the separation of different types of layers. And now we see actually the gel electrophoresis. Okay, in gel we use the different type of thing. First of all, the power supply we use to provide the voltage and the time. Okay, in this, if the voltage, yeah, actually the voltage and time are inversely proportional. Okay, in which if we give, if we increase the voltage, the time will be decreased. Okay, by voltage, by increasing the voltage, the speed of moving the uh, moving the bands will be. And as we say, voltage is inversely proportional to the time. Okay, if we increase the voltage, the speed of bands will be increased. Okay, and the time it takes to move will be decreased. So ideally, we use 90 to 110 voltage and almost 40 to 60 minutes. Okay, now in normal condition, if we increase the if we increase the voltage and also we increase the time of the gel, what will be happen? I am giving you the question. You can answer me after in the question and answer section. If we increase voltage and time in gel electrophoresis, what will be happen? And okay, think about it. I want to get the answers in question and answer section. Okay. Actually, the longer bands means it will be heavy, should be heavy bands, and the shorter bands it is the lighter bands. The lighter bands move fast, okay, and the longer bands move slowly, okay, because they can't move through the tail, but the shorter bands can. Okay, so that is why the section of the movement is that, and longer bands are close to the valves, and shorter bands move far from the valves. Okay, now we'll start from the sample collection first of all. So in the sample collection, we collected the mostly we use the golden. You can say the golden source of sample collection is the human blood. Okay? And we can also collect the sample from soil like that, and we also can collect the like the microbacteria collecting from bones. So these are the pipers, tips, like okay. So what is the purpose of collecting the sample? First of all, the obtain the biological material. Okay. In every sample we collect, we actually try to obtain the biological material. That is DNA or RNA, maybe also the protein. So sources, the main sources are human, like human. So in human, we can collect the blood, we can also collect the saliva, we can also collect the tissue biopsies. Okay, the mostly the cancer cells and the hair follicles. Okay, now our hair follicles also have it here. So can we use? Uh, okay, I have a question. Can we? I have a second question. Can we collect wet for DNA or okay, I want give you a second question. You can have question and answer section. Okay, in animals we also collect blood, air punches. By air punches we also get the blood and the tissue samples. The tissue samples I like the tissue biopsies. And in plants we mostly collect leaves, seeds, and roots. In microbes, mostly use the cultures and environmental samples like that, like the soil or the pond. after collecting the sample, we preserve it or we directly collect the sample, obtain the biological sample like DNA or RNA. In preservation methods, we can snap freezing in liquid nitrogen, okay, at minus 190, 190, 60. And using RNA stabilizing agents like RNA letter. We use RNA letter that can actually stabilize the sample. And storage in buffer solution like TE buffer for DNA. And we can store also in freezers for short term. We can store at 4 degrees centigrade or at minus 20 degrees centigrade. For long term storage, we can use minus 80 degree or liquid nitrogen. Okay, if you have any question, you can give me question. I will definitely answer about it. So you have 5 minutes break. I want the answers about. Okay, I get answer. If we time and voltage, then bands will move faster, but it can damage our result. Okay, and fat itself have no DNA, but fast could have. Okay, Hiramid, uh, you give me a correct answer. That itself have no DNA, but scratch skin could have it. Okay, if someone have a dry skin, they can actually can rub the skin, and when they rub the skin, actually they remove actually their DNA. Okay, and Saira, how much your answer if we increase timer voltage, then bands will move faster? Yes, the bands will move faster, but it can damage our result. Definitely, it can damage our result. Actually, the answer that is very close to it is that when we increase the voltage and time, the gel will be heated. Okay, when gel will be heated, it can make smears in the bands. Okay, so our result will be damaged. Okay, the heating problem by increasing voltage and time, it can damage our results. And what is the solution? If we want to increase, it is if it is necessary to increase the voltage and time, then we should use ice in the gel caster. Okay. And the answer of second is can we collect sweat for DNA? No, we can't collect sweat because sweat doesn't have DNA. Okay, the question is how long can we preserve extracted DNA? Actually, we can preserve DNA for 4 years. Okay, for 2 to 3 years we can preserve extracted DNA, not RNA but DNA, because RNA can be damaged. Okay, because can be degraded because RNA is a single stranded is actually a single stranded or it can be degraded. But if we convert the RNA into cDNA, it can also preserve almost for uh, 2 to 3 years. Okay. Uh, 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 how long can we preserve extracted DNA? It can preserve for 2 to 3 years by uh, liquid nitrogen because at minus 196 degrees centigrade. If you have minus 80 degrees centigrade freezer, you can preserve for almost 7 to 8 months. Okay, there is a second question. If nano drop is not available, how can we optimize an electrolyzer? Okay, you are asking about the primer optimization. Okay, if I'm not wrong, tell me, do you ask me about primer optimization? Okay, okay. You are asking about the primer optimization. If nano drop is not available, how can we optimize? Okay. At which point you want to use nano drop in primer optimization? I think so. In nano drop, it's not necessary to optimize the primers because when you have primer, you can directly use the samples or the cavity edge, or you can use you can use sample the control and the actual samples, and you can use gradient PCR, or the second option is you can use touchdown PCR to do the primer optimization. At which level you want to use Nandro? Kindly tell me. Rahmir, kindly tell me at which point in primer optimization you want to use Nandro? Maybe at DNA extraction level you can use nanodrop, or at DNA level. But after this you can use you directly use PCR, and then after PCR reaction you can use gel electrophoresis. Okay, you at start of PCR reaction. No, before PCR you use nano drop for checking the quantity of DNA extraction. Okay, the extracted DNA, not the primers. Okay, for primers you don't want sorry you if I am not wrong, for primers nanodrop is not used. Okay, for extracted DNA or RNA you can use nanodrop. Okay. Yes, if you want to use the quantification, you can use after PCR. After PCR, you have to run the gel and see the result. Okay, if anyone has question, kindly ask me. Welcome, welcome. If you have any question, you can ask me. After 30 seconds, we will start the next part of the session. Okay, let's start the next part of the session. I am going to share you the screen, all of you. Do you see the screen? Kindly tell me in the chat box. Can you tell me, do you see the screen? Okay, Hiram is see the screen. but the Saira, Saira, I think so. You should leave the session and then join it again. Maybe you have a problem in your device or in the joining. I think so. Okay, start the part or the next part. Okay, now we will talk about the uh, nucleic acid extraction, in which we talk about DNA and the RNA extraction. So, in the nucleic acid extraction, so what is the purpose of nucleic acid extraction? Actually, we isolate the pure nucleic acid free from proteins, lipids, and other components. Okay. Actually, we have DNA in the cell. In the cell, we have like this. That is the thing. So the cell membrane that is made of protein, the made of like uh, made of lipids and other contaminants like carbohydrates and different types. Okay. So we want to break it. We also want to break the nucleus and we want to take that DNA. Okay. So other than the DNA, all are contaminants. Okay. We want to extract the DNA from the cell. Okay. We want to take the pure DNA. So how can we do this? By doing the nucleic acid. Okay, if anyone want to join. Okay, there are different type of methods. One is the manual method, the other one is the automatic. Okay, in manual method we use the phenol chloroform extraction. That almost takes 2 days, and the second one is the trizo reagent RNA extraction that almost take 1 day. I love that method, RNA and phenol chloroform extraction because uh, it, they give the very good concentration and the purity. In automatic methods, it is also good if you want to get the result within hours. Then you can go with automatic methods, in which we use silica column based kits and magnetic field based verifications. Okay. What is the pros and cons of manual methods and automated methods? I want to ask you. Yes, Mazhar, recorded lecture will be provided to you in the WhatsApp group. Okay, the link will be provided to you to all of in the WhatsApp group. So if you feel any network issues, you can leave the session. Okay, I have a question. Can you tell me the pros and cons of automatic uh, both manual and automatic? Okay. First of all, talk about the DNA isolation extraction. Okay. Uh, uh, the protocol will be provided to you in the WhatsApp group. Okay. So you don't need to worry about that. Okay? Actually, we talk about the concepts actually behind the DNA. Okay, and there are three general steps involved in DNA isolation. Okay, first one is the lysis. We lyse the cell. I have a cell. I have a cell membrane, the nuclear membrane. Then we have the queen, the DNA, or the place that is actually found in nuclear membrane. We want to extract that, the DNA uh, from the prison of cell membrane and nuclear membrane. So first of all, what is the step to do that is the lysis. We lyse the cell. Okay, we lyse the cell. We break the cell. Then we remove the contaminants. Okay, then we remove all the contaminants. After lysis, we remove the contaminants, and at the end we recover the DNA. Okay, then we recover our queen DNA or the prince if it is male prince. Okay, in lysis we use SDS. So what is SDS and why we use SDS? We mostly use that name, the SDS, the protein K, and the PCI, that's from Y, and that's propanol and the chalprin, ethanol, and different types. Why it is used? You should know about the why, what is behind it. Okay. If we talk about we use PCR, why? We set the temperature at 95 degrees centigrade. Why? Actually, the answer of the why is more difficult. If someone asks about why, we actually quarrel with him. Actually, in this manner, because we don't want to give the answer of the voice, but we should know about the voice. SDS is used for ion balance, okay, to maintain the integrity of the DNA cycle. When we lyse the cells, there is a definitely there is definitely a chance we can disturb the pH of the cells, okay, because in the nuclear membrane the pH is maintained, okay, because the cell is covered, the pH is maintained. But when we lyse the cell, then there is a definitely chances we can change the pH of the cells. So for ion balances, for maintain the pH, we use SDS. Okay, we use SDS to maintain the integrity of the sample, so the DNA should not be degraded. So the next thing we use in the lysis is the protein is A. The word tells us they actually degrade or the denature the protein. Okay, they actually break down the protein. So the breakdown proteins, even those present in the nucleus and cell membrane. Okay, cell membrane is actually made of protein. Nuclear membrane is made of protein, so they can break down that protein. So the next part we use the next step in the DNA isolation extraction is the removal of contaminants. So we use PCA, no chloroform isoamide. So in PCI, it is actually used to phase separation. PCI is light sensitive, so we wrap in aluminum foil. So what is phase separation? So the basic function of isoamide in PCI is to prevent foaming formation. Okay, when we do the phase separation, the mostly thing occur is that there is a foaming formation on above the DNA or the between DNA or RNA. Okay, that can contaminate our DNA. So we use isomite to prevent that type of forming. And at the end, we recover the DNA by using isopropanol that actually precipitate the DNA. They make the pellet at the end of the solution. Okay, at the end of the solution. That So we can remove the solution, and but we have DNA at the end. And child ethanol is used to remove the remaining contaminants. If any contaminants is present, can be removed by child ethanol. So this is actually the concept behind the theory of DNA. I am here to give you the whys, answer of whys. I am here to give you the concepts of the molecular biology. I'm not giving you just the theory, but actually give you the actual concept behind it. So this is the simple uh, diagram in which this is the tissue biopsies like sample. Okay, we homogenize it. We use lysis. Okay, by using guanidinium, you know. Then there is the RNA, protein, DNA. There are different type of things in the solution. Okay, we need DNA. Okay, so we do the phase separation. In the phase separation, you can see the contamination is in the bottom. Okay, and the above one is the DNA. Okay, sorry, there is the RNA pellet. In the RNA, same thing happened, but you can assume there is a okay. So above one is the DNA. Then we use isopropanol and we precipitate the DNA at the bottom, and the above one can be removed. And then we use water or the nucleus water to resuspend. Why we resuspend it? Because we want to take it for our next steps. I know someone might. Hmm. Now, if you have any questions about nucleic acid extraction, you can ask me here. If you have any question about nucleic acid extraction, you can ask me here. Okay? The DNA and RNA extraction is similar. Okay? It's actually the same. I will provide you the protocols in the WhatsApp group. Now, if you have any question, you can ask me in the chat box. Okay, I get the answer from Saida Kramer. The question is actually can you tell me the pros and cons of manual and automatic methods? In manual method has low cost, okay, but time consuming. Definitely, Saida Kramer, you have a good knowledge. And automated method is accurate but expensive and requires technical skill. Okay, Saira Ahmed, the first line you give me the answer is definitely correct. Okay, manual method has low cost but time consuming. Okay, and the automated method is accurate and but expensive. So it is correct and require technical skill. Actually, uh, the automated method is very easy. Okay, if you if you ask the children to do it by providing by guiding it for 5 to 6 minutes, they can do it. Maybe the handling is different. Maybe we get the concentration or the purity is somewhat low, but they can do it. Okay, so there is no required the technical skills like in the manual methods. Okay, but in automatic methods, the concentration is very low. The DNA or RNA we get, the concentration is very low, but the purity is high. Why? Because the steps we would do in the automated method is very less. For log, like you can do just 5 to 6 steps. Okay, so the purity we get is very high, but the concentration is very low. In in the manual method, because the steps of the DNA is almost 25, okay, and it takes almost 2 days. So the steps are very high, and the purity we get, the chances to get the purity is very low, because the handling and the different type of contamination can be done by the students or by the surgeon. So the chances to get the purity is very low, but the concentration we get is very high. Okay, it means we should use more concentration of DNA. We use what? Uh, Hira, actually, we we don't know the concentration of DNA, because we have blood. For example, we have blood. We I am not know the actually what is the concentration of DNA, because we have to just extract the DNA. Okay, do you got get it? Rahvi? We don't actually at that point when we extract the DNA, we are unable to tell that how much concentration we get. After getting the concentration, we can tell about it. Okay, Hiram, we have question. Can we use boiling method for bacterial isolation? Um, Bacterial DNA for bacteria. Yes, in we use the culturing method. Okay, in culturing method, at some we have a different type of steps. Uh, the 1 to 2 steps at that point we use the heating process. Okay, we use heating to kill the bacteria and extract the DNA. Okay, so the boiling method can be used for bacterial DNA as well. Got it? I will share you the protocols of thermoscientific datametric methods of the DNA, RNA to you in the group, in the WhatsApp group. You have 2 minutes. You can rest. You can ask me any question, or you can just rest. Okay, the answer we get from Hiramid and Saira Kramat about is Ethereum bromide. Very good. Hiramid, the question is band don't open, how long can we run the gel again? I, I think so. You ask about the if the band actually the band is not open, it is actually due to if your handling is poor. Okay, if you handle the gel not well, then band will not be open. And the second thing is if your DNA is degraded, okay, or DNA concentration is very low, okay, then band will not be open. How long can we run the gel? You can run the gel if you don't get if you use the loading die. Okay, you use loading die to see where the actual DNA band is. If you see the band is not open, you can run the gel how much times you want. Like you can move, you can run the gel for 1 hour. Okay. If so, you can run the gel definitely for infinity. If band don't open, if band open, then you are limited. Then you should run the gel until it doesn't cross the gel and move into the buffer. So Saira Kamath also have a question. This question is from previous topic. Okay, no problem. But I am confused about this. If CDN is also derived from RNA, then why don't we use extract RNA directly? Why do we create CDN instead? Okay, mm, Tarak Raman, if uh, I got the question. If I didn't got the question, you can ask me in the chat box again. I think so. You want to ask me that why we create CDN? Okay, why don't we use RNA directly? Okay, actually, RNA just have uh, manner to ask the question. Okay, uh, let's assume a scenario. You want to check the gene expression. Okay, then you need to check it by using RNA. You can't do it by using DNA because DNA tells about the genomics, about it tells about the what is actually is it heterozygote, the homozygote, and that type of thing. Okay, it doesn't tell us about the gene expression, but the RNA tells us. Why? Because RNA is actually derived from the DNA. Okay, if RNA is created, it means DNA works. So it tells about the gene expression. So why don't we directly use RNA for that? Because because qPCR just needs cDNA to do that. Because cDNA is actually a double-stranded DNA. Okay, it is complementary to the DNA. RNA have uracils and it is single-stranded RNA. If we directly use RNA for qPCR, it will be degraded and we don't get any results. Okay, then we need cDNA to do that. Also, I give you the answer. If you don't get the answer, you can ask me in the chat again. I will answer it more elaborately. Okay, Sarah Grammar, thank you so much if you got the answer. Okay, Hiramir. Okay, the session is over. Okay, and thank you so much. I will definitely see you again in tomorrow's session. Hiram, we have a question. Please let us know if you need any software for primer design for tomorrow's session. Hiram, there are different primer designing softwares. Okay, but now I want to tell you that primer 3 you can open the primer 3 for tomorrow. Okay, tomorrow you can open the primer 3, but I will explain the all things in primer 3 like things in the session. Okay, you don't need to do anything, but I will encourage you to do the practical things after the session. Okay, like I will share you the protocols and you can see it, and you can I will definitely encourage you to do all things with clarity, with concept, not just mix the things, but you also know what is actually we do, why we do. Okay, if we pray for years, okay, prayer means for years, but we don't know why we do that. What is uh, actually the funda behind it? We, if we don't know why we pray, if we know I want to happy our God, then we need to pray. Okay? And we need Jannah to heaven. If we know that, then we pray. If we know the why we do that, then we definitely have interest to do that. Thank you so much for being part of this wonderful journey. Follow the further workshop announcement and a lot more. You can follow me at LinkedIn, Zynome Scientific, and you can also connect with me at LinkedIn by Alidras and I have youtube.com Alidras. You can see my sessions also on YouTube. If you have any question, you can ask me by emailing or by just through WhatsApp. Thank you so much. You can leave the session. Thank you so much. Assalamu alaikum. Welcome back to the workshop. that is actually the international online workshop on molecular biology techniques. This is the second day of our workshop. So this is the session, it's actually the last session, or now I am going to share you the slides to you, and let's start uh, from our next part of the day. Okay, today we will talk about actually the things you already know about it, and I am very happy that you actually mean the workshop and give me a very positive response about the workshop that it is very insightful. So thank you so much because your suggestions and your actually the preference to give me to work hard is very important for me. So thanks so much. So let's do this with dedication. Okay, now as you know, in the first day we talk about the sample collection, then we talk about the nucleic acid extraction, and we talk about DNA and RNA. Then we talk about quantification, in which we talk about how we can quantify our extracted DNA by using different type of techniques like gel, electrophoresis, and nanodrop like spectrophotometric. And we have a very interactive session in which we ask questions, you ask the question, I give the answers, so you give the answers. So let's start the second day of our workshop, then in which we talk about the PCR. So what is the PCR? And then we talk about the real-time PCR, then we talk about the primer design. Okay, so and at last we talk about application. So first of all, we talk about the polymerase chain reaction, in which we talk about how we can amplify our DNA. Okay, so the word polymerase actually is the word of it is actually the name of an enzyme. Okay, it is actually an enzyme. It is actually an enzyme, and that what can do this enzyme is actually is polymers. Okay, it can add the polymers to each other. Okay, when the DNA phase we have the attachment and the extension of the nucleotides to make the DNA is done by polymerase. Okay, and the word chain reaction is actually that it do by doing the cycles in cycle. Okay, so it they do one. Okay, that two, then it makes it four, then makes it four, six, eight. Okay. First of all, 2, then 4, then 8, then 16 in the exponential. So this is the exponential reaction. There is a chain reaction. This is going to use the polymerase. So what is the principle of polymerase? It is a technique used in molecular biology. It's a technique, okay? In vitro. The word in vitro, it means that we take the blood outside from the body. Then we do extraction. Then we do extraction, and at last we do PCR. Okay, so the blood we take, it is actually outside the body. It means that in vitro. Okay, not in vitro. A DNA polymerase used. Okay, in vitro we use DNA polymerase that is used to amplify a piece of DNA. Okay, it is actually a gene of interest. It is actually that gene that is interested by us. Okay, that gene of interest we use to amplify. by enzymatic replication. Okay, actually, the replication occurs in our body also is an enzymatic replication. Okay, in which the different type of enzymes are used, but in polymerase chain reaction, we use enzymatic replication by using DNA polymerase of different type of polymerase. Okay, not actually the polymerase that's used in the that is actually produced or used in the human, but we use the tag polymerase that can be bear the denaturation step like at the next. Okay, the DNA generated is used as a template for the lab. Okay, the DNA generated. Okay, first of all we have this type of, sorry, the DNA is uh, the denature, and after it new DNA is created. Okay, and the old DNA is used as a template to generate another DNA. Okay, that is why the word used the DNA generator is a chain of reactions. Okay, there is a reaction chains. Okay? There is a cycle. Start in which the DNA tablet is exponentially amplified. Okay, exponentially amplified the two, then it makes four, then eight, then we can say 16, then 32, and so on. So it amplifies even a single piece of DNA. Okay, a single piece of DNA to millions or more copies. It can do. This is actually the brilliancy of the polymerase chain reaction. So it employ a heat stable DNA polymerase. Okay? They actually use a DNA polymerase. That's the heat stable, such as like the tag polymerase. Remember it? It used tag polymerase. An enzyme is isolated from bacterium thermus equity. DNA polymerase enzymatically assembles a new DNA. Okay? DNA polymerase enzymatically assembles a new DNA. Okay? It's a very simple. Single-stranded DNA used as a template. Okay? In the previous slide, I give you the concept that the single-stranded DNA. Okay, first of all, this is the DNA. Okay, the DNA chain, this DNA chain, and this one is used as a template to create another DNA. So single-stranded DNA used as a template, and DNA oligonucleotides like the primers required for initiation of the DNA. I am going to use a different type of marker like this. This is the primer. The primer is used to initiate the initiation of the DNA. Okay, Saira Kramak, kindly tell me, do you see the screen?
Yes,
Okay. Just mute your mic now. Okay, thank you. Uh, actually, I have started the today's session. Okay, if you miss the first part, you can see it in the recording. Okay. Uh, and then it actually use the DNA primers to initiate. So it uses the thermal cycling. I will tell you in the next slide what is actually the cycling. So what is the requirement? Okay, I will tell you the thermal cycling. Actually, in PCR, we use different type of heatings. Like first of all, 95 degree centigrade. Okay, at which we do denaturation of the DNA, in which the double stranded DNA is denatured into sorry, double stranded DNA is denatured into single stranded DNA, and the primer is attached with it, and the new tablet is sorry, a new strand is rejected. So at 95 degree centigrade, it is done. Then then at 72 degree centigrade, sorry, at 55 degree centigrade, annealing is occurred. At annealing, actually, the primer is attached with the template. And then at 72 degree centigrade, it is actually the optimum temperature at which extension is occurred. So that is why the word is used here. It uses thermal cycle. So what are the actually the requirements of PCR? First of all, we have DNA template. Okay, a DNA template is used. DNA template require having the target region. Okay, the DNA template should have the target region. Okay, what is the target region? The gene of interest. Actually, the that gene that is interested by us. Okay, in which the you think the problem is occurred. Okay, or we want to check the information of that part of the template. Then we need two primers. Okay, the first one we use the forward primer, and another one is the reverse primer. We are in the requirement we have the two primers. Okay, the third one we need DNA polymerase, such as tag polymerase. Why? Because the extension of the new template is done by DNA polymerase. And no another one is the TNTP. Okay, TNTPs are used to extend the new template because they actually add add the template. Like this one is the template and this is the primer. So TNTP is attached here to extend the one new strand. Okay, DNA polymerase actually add. So that is why the new strand is created. And definitely the buffer solution because they actually maintain the pH. And the divalent cations, we actually need the divalent cations because in PCR, the salt concentration, the salt and temperature, the two keywords, salt and temperature should be optimally maintained. What word we use? Optimally, optimally maintained. Why? Because if we increase the salt concentration or we increase the temperature, the sample will be degraded or denatured. Okay, so the new tablet will not be created. So it should be optimally maintained. The volume used for PCR is 20 to 155 microliter. Someone says 70 to 150 microliter. Someone say 20 to 150 microliter. It should be in between. Okay, because if the sample is very low, the sensitivity of the conventional PCR is just 20 to 155. If the sample is less than 20 microliter, the sensitivity is very low of PCR. It can't detect that. Okay, so the reaction will not be occur. If if it is if you run the sample, we don't give any results. The volume used for PCR is 220 to 151. In small reaction tubes, okay, we use the PCR tubes like very small tubes we use for it, like 0.2 to 0.5 milliliter volumes. Okay? Placed in thermal cycle. Okay, we use the thermal cycle for it. The thermal cycler heats and cools the reaction. Okay, first of all, 95 degrees centigrade for denaturation, then 55 degrees centigrade for annealing, and then 72 degrees centigrade for extension, and then at hold. So what are actually the steps of PCR? Okay, three major steps is the denaturation at 94 or 95 degrees centigrade, then annealing at 55, 54 degrees centigrade. Actually, we do primer optimization for annealing temperature. Okay, so primer. Why? Because primer should be attached with the DN temp. Okay, so the annealing temperature should be okay. Please write it on your notebook. Annealing temperature should be 3 to 5 degrees centigrade less than sorry 3 to 5 degrees centigrade less of the TM melting temperature of the primer. Okay, when we design the primer, we got the TM of the primer. Okay, for example, we got 57 degrees centigrade. Okay, the annealing temperature should be 3 to 5 degrees centigrade less. So it should be 54 degrees centigrade or 53 degrees, at which is they give you the best results. Okay, so you can select extension is always at the 72 degrees or near to it, because it is actually the optimum temperature to extend the to extend our gene of interest, which are repeated for 30 to 40 cycles. Okay, for 30 40 cycles that are enough for making the millions and millions of copies of your uh, amplicon on an automated cycle. Okay, so you don't need to stop your PCR, then add new things in it, or you have to not stop at any cycle. Okay, so this all can be done within 1 to 2 hours without your control. They can heat and cool the reaction mixture in a short time. Okay, like 1 to 2 hours. Your reaction will be done. So again, these are the steps. First of all, 1 minute at 94 degrees centigrade. Okay, your double stranded DNA is converted into, it will be denatured into single stranded. Okay, then at annealing, that is only for 45 seconds for short centigrade, at which like that one, the forward and reverse primers attached with it, and that extension. So the extension is occurred. Only D and DPs are used. Like you can see here, first of all denaturation, then annealing, and then extension. Okay, you can see the extension can be occurred at 54 degrees centigrade. Okay, but the optimum temperature need to extend our new strand is at 72 degrees centigrade. So detection of PCR product. How we can detect the PCR product? To check whether the PCR generated with DNA fragment amplimer, or we can also say amplicon. Agarose gel electrophoresis is employed. Okay. Ag- Sorry. Agarose gel electrophoresis is used to check whether the PCR generated the anticipated DNA fragment. Okay. For the separation of the PCR product, we do gel electrophoresis. Okay. Just remember it. We use agarose gel in convention. In real-time PCR, we don't need gel. The size of the PCR product is determined, okay, by comparison with the DNA ladder. We use DNA ladder, that is the molecular weight marker, which contains DNA fragment of known size run on gel alongside the PCR product. Okay, so in gel, what we do? First of all, we do the vortexing just to mix our DNA. In, in, okay, sorry, our amplicon. Then we do PCR. After PCR, we take the sample and we load the gel, load into the gel. Okay, that loading. After running the gel, we see here like that type of things will be occurred, and then we can check in all gel doc. Okay, we can see all on gel. So what we get actually? So this is the ladder. So at that point, it will be 500 base pair, 400 base pair, 300 base pair, 200. The PCR product, like the three samples, is actually is very close to the 500. Okay, so it will be 484. Yes. So how we can detect the PCR product. So if you have any question about PCR, you can ask in the chat. You have 5 minutes. If you have any question, you can ask in the chat. Okay, Siren, you have a question. What would happen if the primers are not specific? Okay, if primers are not specific, okay, the non-specificity will be occur and primer will be attached at somewhere else. Okay, it will not be attached with your gene of interest. Okay, so that is why you got a different results. If you got the different results, uh, you will not be you not get what should be in your it should be your hypothesis. Like you want to check that in cancer, that gene will be expressed, okay, or highly expressed in that case, okay. But when your primers will not specific and they attach somewhere else, and you got no result, okay, and that results give you that uh, that assumption is that that gene is not highly expressed or not expressed in that cancer. Okay, you got 360 degrees and so you can say 360 degree reverse answer is answer. Okay, so primers should be very specific. So the primer dimers will not be occurred. There will be not smears and there will not be any difference in your PCR product. Okay, so that is why primer should be specific. Okay, the another question you have is how we select the primers specifically. Okay, we do primer designing for this. Okay, first of all, you get the sequence. Okay, when you get the sequence, then you make primers within that sequence. Okay, and when you create the create primers within that sequence, then the whole genome in the whole genome the primers only uh, will only select at that point of your sequence. Okay, will only sit at that point. Okay, so the primer designing is very important. It's very important for your reaction, for your whole research primer design. If your research is on molecular biology and is all on and PCR is used in it, then whole results are depend on primer designing, or are depend on your dry lab, all depend on your bioinformatics. Okay, so that is why primer designing is very important. Also, you got the answer. Okay, thank you. So let's start the next part of our session, which is real-time PCR. I am going to share the screen to you. Okay, real-time PCR, the principle is exactly same like the conventional PCR. Okay, but in real-time PCR, we got the real-time monitoring of the application. Okay, what is actually done in your reaction is seen by your eyes. Okay, that is the real-time monitoring of the application reaction. Now, in conventional PCR, you got the answer, you got the results at the end of the at post PCR by doing your gel. But in this, gel is not required. Okay, so that is why it is very good and it is advanced. The definition of real-time PCR is that real-time monitoring of the application reaction. Very simple. Real-time monitoring of your real-time monitoring of the application. So that is the real-time PCR. What is the purpose of this? To estimate the initial quantity of specific entity. The, the main reason. of the real-time PCR is that it gives you it gives you the initial quantity of the specific stability DNA. Like at start, you go to okay 200 microliter of your 200 nanogram concentration of your specific temperature. Okay, or like 200 base pairs of your temperature. Okay, the initial quantity of the DNA can be estimated by real time. So what is the principle of real time PCR? It's very simple, and it is exactly same in like the conventional PCR. So what is actually difference? The sensitivity. It is very sensitive. Okay, in conventional PCR, it should be 70 to 150 base pairs. Okay, in this it can be 20. Okay, so sensitivity is very high in real time PCR. It is very specific. Conventional PCR is also very specific, but real time PCR is more specific than the reproducibility. Okay, the first in first reaction you got 70. For example, you got the fault in 70. In second, you also get the same result 70. In third, you got same result 70. Okay? So the reproducibility is very high. Okay, so that is the sensitive, specific, and reproducible quantitation. Okay, quantification of nucleic acid. Fluorescence is measured every second. Okay, in real-time PCR, fluorescence is occurring. Okay, we use different type of dyes or the probes in real-time PCR. So that is why the word is fluorescence. Fluorescence is measured in every cycle. In every cycle, fluorescence is produced. Okay, if the fluorescence is very high, okay, then the curve will be upside, and that gives us the answer that amount of PCR produced is very high. Okay? So that is why they are directly proportional. And the detection of signals during early phases of reaction. Okay, we can detect the signals at the very early. Okay, like that. At that stage, at that you can say at no phase, at which the exponential increase of your DNA is not occurring. Okay, at that stage, the primers are actually searching where to attack. Okay, when they know it that there is actually the point where we have to attack, then the exponential phase starts. At that phase, DNA tablet is exponentially copied and increased. Curve rise. Okay, the in that phase you can also check the theory of my concept is that curve rise after a number of cycles. Okay, after how many number curve rise? Actually, after 8 cycles. Okay, after 8 cycles, you got the exponential change. Curve rise after a number of cycles. After a few number of cycles, that is proportional to the initial amount of the. Okay, that is proportional to that. For example, at first you go 2, then 8, then 32, then 64, and so in exponential. So what are the advantages of real-time PCR? Amplification can be monitored real-time. Okay, first of all, this real-time monitoring is actually the advantage of real-time PCR. Wider dynamic range of up to 1 to 10 fold. Okay, we got different type of results. Okay, so we can measure that change. In gel, in conventional PCR, we do gel. Okay, so the wider range measurement is very difficult. Okay, but in real time PCR, you got the numerical values, so you can check it. You don't need to even analyze it. Okay, so the human error is very low. Results are expressed as you see that as I told you later. I have told you that results are expressed as numbers, okay, in variables. Okay, so due to numericals, the human error is very low. No post PCR processing of your product. Okay? So no gel-based analysis at the end of it. Okay. So these are actually the advantages. Ultra rapid cycle. Okay. In 30 to 2 hours. Actually, this one is the conventional PCR also do that. Highly sequence specific. Okay. Conventional PCR is also highly sequence specific and sensitive. Okay. The sensitivity of real-time PCR is very high. Can detect as little as two-fold change. Okay. Even two-fold change can be measured by. Okay. Two-fold change means that 200%. Means that if there is 100, then if there is change is 200, okay, then it means that there is a double change in it, okay. That can also be measured by real time. So what are the disadvantages of the real time? So it requires expensive equipment. Equipments are very expensive, even in millions or millions rupees. Reagents and the conventional PCR are the same, but the master mix used, the cyber degree master mix used in the real time PCR is very expensive. Due to its highly sensitivity is very high. You may get high deviations of these same experiment. If you reproduce your when you do repeats in your experiments, you can get deviations because the sensitivity is very high. Maybe contamination will be occur. Thus, the use of internal controls. Okay, you have to use the internal controls like you can use gap DH. Okay, gap DH can be used. Internal control. That that is very recommended in gene expression experiments. Okay, so then you can get your results and that are very specific. Okay, if you have any question about real-time PCR, you can ask me in the chat box. If you have any question about real-time PCR, you can ask me in the chat box. You have 5 minutes. You can search about it and you can ask me in the chat box. You have 5 minutes to rest or to ask me any question. After this break of the question and answer section, we will go towards primer design.